Hello, everyone. Welcome to the ELSC seminars. Today we are hosting Professor David Hansel, who is a senior researcher at the CNRS Paris. David did his PhD in theoretical physics at the Marie Curie University in Paris, and then he did his postdoc training here at the Hebrew University, again in theoretical physics. Today, David is a senior researcher at the CNRS at the Center of Neurophysics, Paris Descartes University, and is also a co-director of the France-Israeli Laboratory for Neuroscience. David's studies focus on theoretical neuroscience, where he tries to model how emerging properties such as orientation selectivity arise from the connectivity of neural networks. And today he's going to talk about the theory of feature selectivity in rodents' primary visual cortex. So before we start, please make sure that you are all in mute mode. You're allowed to ask questions during the talk, but there will be also time for questions at the end of the talk. And I welcome David to step in.
Thank you very much, David. So it's really a pleasure to give this talk and using this fantastic technology, which is with Zoom. So today I'm going to talk about a project that together with my colleague Carl von Vreeswijk, and we started, it was more or less 11 years ago. It has to do to develop a theory of orientation selectivity in mouse V1. And I have added here additional claim outcome of the talk, which is why mice are not cats. So the menu, the outline. Okay, now you have to. Okay, so first of all, let me acknowledge my collaborator in this. So for the theory, it's Carl von Vreeswijk that I already mentioned, and Herman Mato from Argentina. And there will be also, I will report some experimental results, which were obtained by our collaborator, Nicolas Privé, and is former PhD student, now postdoc, Jagroti Patatkar. So the menu of the talk is as follows. So I will make an introduction, which entitled From Cats to Mice, which also we do two things. One, so that we are all on the same page, and also which will us to formulate the problem, the hypothesis that we're making, and about this problem. And this will serve as an introduction to the second question, so the second part of the talk, which is how layer 2-3 can inherit orientation selectivity from layer 4 under the hypothesis that we are making. So this is assuming that there is already orientation selectivity in layer 4. So it's really inheritance of orientation selectivity from layer 4 to layer 2-3. That's natural question which comes in is, now we would like to know why there is orientation selectivity in layer 4. So this is a question of the emergence of the orientation selectivity in layer 4. And here it will be divided in two steps. The first step will be to understand, to describe the structure of the thalamic excitation. So the excitation coming from the thalamus, from the LGN to layer 4. And the second step will be to understand the consequences of this thalamic excitation on the visual response property in layer 4, and to show that indeed selectivity can emerge in layer 4 under our hypothesis that I will make later. And then we'll arrive to a prediction. Our theory leads to a prediction. Then the question is, so I will formulate the prediction, and then ask the question whether it's falsified or verified experimentally. And if I'm asking the question, it can anticipate that it is verified, especially because it was published in 2018, and this is with our collaborator, experimentalist collaborator. And at the end, I will ask the question of the role of a few specific connections in V1, because our hypothesis in all the steps from this, the question about layer 2, 3, then inheritance, about the question of emergence, and the prediction will be that, in fact, in mice, in contrast to cats, the connectivity is highly disordered. So there is no or very few specific connections, feature-specific connections, either between, between the neuron in the cortex, and also there is no organization in the projection from LGN to the cortex. So let's start with the introduction. So from cats. So it's well known that there is orientation selectivity in the cat visual cortex. Actually, this was one of the very first paper, Jubel and Wieser. It was the discovery of this orientation selectivity, among other things, that they discovered the visual cortex. And it was one of the first, I think the first, example of selectivity in the cortex. Selectivity to stimulus, or selectivity to action, or selectivity to memory later. Now, what was the experiment? So the experiment, most of you know that. So the experiment was performed originally on cats. So the cat is here. There is recording, so extracellular recording, and the cat is seeing a screen on which there is a light bar stimulus, which is going to move. So which is projected on that screen. And when Jubel and Wieser recorded the neurons in the cortex, so they found that depending on the orientation of this bar, the response of the neuron was more or less strong. So this is taken from their paper of 1959. So you see that this neuron responds preferentially when the bar is oriented in that direction. And when the bar is oriented horizontally, so the response is much, actually there is no response. Okay, so this kind of selectivity can be quantified, or first of all, can be summarized in what is called tuning curve. So in this case, and in most of these, this is coming from a paper from Anderson and collaborators. This is from CAT. In this case, so the visual stimulus was a drifting rating. And this is very important because in all the talk, the visual stimuli that I'm going to consider are drifting ratings. So drifting rating is characterized by the orientation, by a temporal frequency, and a spatial frequency. And the spatial frequency will be a particular, very important parameter in the studies that, in our theory. There is a wave vector, which is a vector which is perpendicular to the motion of the, or to the drift of the grating. And theta, so is the feature that the neuron is going to encode. And this is a tuning curve for different contrast measured. So this is a typical best because there is a lot of, in fact, diversity. But this is so this shows quite, a substantial, strong tuning. And as a function of the contrast, the peak increases and the width does not change much. The preferred orientation is defined in the orientation of the stimulus at which the response is maximum. So in fact, if one rescale this normalized the tuning curves at the maximum, at the peak, so we find that for this neuron, there is almost perfect tuning invariance, so the preferred orientation does not change with contrast, and also the tuning does not change with contrast. The bandwidth of the tuning curve after scaling does not change with contrast. Okay, now there is another invariance to the feature of the stimulus, which is the fact that the preferred orientation in cat, So this is for cat, this is for monkey. The preferred orientation does not change with spatial frequency. So this is from this study from 1985. So this is one neuron. These are the tuning curves at different spatial frequency of the grating. And what you see that the preferred orientation almost does not change. So this can be also visualized in that way. And here we see what happens for many neurons. So this is from monkey, actually an earlier study. And here, so it's a drifting grating. So for a stimulus which can have different orientation and different spatial frequencies. So this is the vector, the wave vector, depending on the length of the wave vector, depending on spatial frequency, the length of the wave vector is more or less large. Now, what you observe here, so here, all these regions which are gray correspond to the combination of spatial frequency and stimulus orientation where the neuron is active. So now what you see that these regions are elongated and they are along the radius. So which means, and it goes to the center. So what that means? That means that the preferred orientation of the neuron does not change with the spatial frequency, does not change with how far we are from the center. This is true for this neuron, this is true for that neuron, this is true also for this neuron, etc., etc. So this is the second invariant. So in a way, what we see here is that there is kind of decoupling between two information channels. Between three information channels. One, which is orientation, second, which is a contrast, third, which is a spatial frequency. Now, these are the very few equations that will be in this talk. So how to quantify? So this is where we are on the same page. This is how to quantify the notions that we are going to use all along the talk. So first way, of course, is by the tuning width, so which will be the width of the tuning curve at half. So half of the maximum. There is a mean response. So which means that here what we are going to do is R of theta k, this is a response of the neuron for stimulus at orientation theta k, and we sum over the difference theta k, and this will give us the mean response, mean over the orientation of the stimulus. There is also the response modulation, which is a complex quantity, so with a modulus and with a phase. So this is calculated as a sum of R of theta k multiplied by this exponential 2I theta k. And we normalize to the number of stimulus, to the number of stimulus orientation. Now, theta is in fact this phase is in fact the preferred orientation of the neuron, and if it takes the ratio of R1, the amplitude of the modulation, to R0, so then to the mean, this gives us a measure of the degree of selectivity, which is called orientation selectivity index, and that we will abbreviate by OSI. So OSI, if OSI is 1, it means that the response of the neuron is only at one orientation. And if OSI is 0, so it means that there is no modulation with orientation of the stimulus. That means that there is no tuning. Okay, there is another very important property of this orientation selectivity in cat and monkeys, and which actually was already discovered by Hubel and Wiesel, so very long time ago. This is the functional organization of primary visual cortex of cat and monkey. What that means? That means that neurons which are close by tend to have the same preferred orientation. And neurons which are far apart tend to have different preferred orientation. So you see that there are orientation domains. And what that means? That means that if the connectivity depends on distance, and neurons which are close by will tend to interact more than neurons which are far apart. So this organization immediately transfers into an organization of the connectivity of the functional connectivity, or more precisely, that the connectivity will be functional in the sense that neurons which are nearby, which interact more, will tend to have, and which have the similar preferred orientation, will interact more than neurons with different preferred orientation. So again, if the neurons which interact more are those with similar preferred orientation, and neurons which interact less have different preferred orientation. Okay. So what is the mechanism? The idea about the mechanism in that case. So it makes use of two components. And in both components, the emphasis is on the organization. So there is, first of all, an organization from the thalamic projections to V1. So they are organized in a way that you know probably, I'm sure that you know that in the thalamus, neurons have on and off subregion. In the on subregion, so they respond to light. In the off subregion, they will respond to absence of light. So the idea is, so this organization is as follows. So you take one neuron in layer 4, and it receives inputs from a set of neurons in the LGN, and these neurons are aligned on the in the visual field. So it means that these neurons, so you see that the center is plus, the pourtour is minus, so this is on, this is off. So a bar which will be oriented like this will elicit more response in the neuron in the cortex than bars which are orthogonal. Now, there is another, so this is the idea that this organization will contribute to the selectivity in layer 4. This has been studied for many years, and it was suggested by Hubel and Wiesel, 1962. So that's why we call this the Hubel and Wiesel mechanism. On the other hand, as I mentioned, neurons which have similar preferred orientations in layer 4 tend to interact more than neurons which have different preferred orientations. So there is more excitation, more recurrent excitation coming from neurons with similar preferred orientation. And this has been shown that this can sharpen the selectivity. Here, there is already some selectivity, yes, from the thalamic projections. Here, because of the organization, and the recurrent interactions will sharpen this selectivity, and this will give rise to the substantial selectivity in layer 4. Now, how much this component and this component contributes? This has been I mean, a lot of works, theoretical and experimental, have been performed in order to to attribute the respective contribution of these two components. So here you have only part of the papers for the experiments supporting this thalamic projection, and for a theoretical paper for studying this contribution of the recurrence. Now we go to mice. So what happened in mice? So first of all, in mice, there is also orientation selectivity. This is known from the mid of the 70s. I think that the first time it was shown was from a group in Lyon, in France, as a lab of Métain. And Métain, and then there were several studies later on. So here I'm taking examples from this study of Neal and Stryker from 2008, and two examples of neurons, one which is sharply tuned and the other which is broadly tuned or not tuned at all. Now, what they show in this study is that the tuning width is comparable in all layers except in layer 5. So you see that in layer 2, 3, layer 4, layer 6, the degree of selectivity measured by the tuning width is very similar. Layer 5, it's less. Now, there is something which is very important. There is here a very important difference, which was already known actually in the mid-70s, that there is no orientation map in V1. So this is called the salt and pepper organization. So you also see that there is also less activity in V1 of mouse than in primary visual cortex of cat and monkey. That's why you see here that there are less neurons which are colored that you had in the previous, in the map of cat and monkeys that I showed before. So the problem is, yes, the mechanism explaining orientation selectivity in cat and monkey, and more generally in species where there is a map, was based on the organization of the connectivity. Now, this leads actually Oki and Reed to make the following statement. The mixed salt and pepper organization of preferred orientation in rodents argues for specific connectivity between neurons. So what that means? That means that taking into account the fact that there is no map, yes, in the cortex. So that means that there will be organization of the thalamic projection, like in cat and monkey. And on the other, and in the cortex, there will be no map. That means that what you see is disordered, but in fact, the connectivity will be specific. So what you see is not what there is. Okay, so again, so in that case, we can use a theory that was developed for cat and monkeys and say, okay, so that's why I say it are used for specific connectivity between neurons. Now, what we are doing, and this is our hypothesis, is that in fact, the mixed salt and pepper organization of preferred orientation in rodents can result from non-specific random connectivity between neurons. Okay, so we mean that even under this hypothesis of pure randomness, it's already possible to have orientation selectivity in the cortex. So in a cartoon, so it means that we assume that there are thalamic projections to V1 are not organized. So these are the on and off neurons from LGN, which is not organized. All these neurons are going to project to the same neuron, to one neuron in the cortex. So there's random convergence of multiple untuned LGN input into layer 4 neurons. And the connectivity is non-specific. So we make the assumption of what you see is what there is. No specificity between the neurons. So which means also that neurons which are nearby, which tend to interact more, okay? So in spite of the fact that they are nearby and they should tend to interact more, in fact, if the preferred orientation is different, they will interact less than those which are far and with a similar preferred orientation. Okay, so non-specificity and projection which are not organized. Okay, and the question is how this can happen, that in such an organization, this is our hypothesis, it's possible to have orientation selectivity. So first of all, the way we are going to proceed is first by considering, so this is the historical, the order in which this project developed. And the first question, I mentioned that layer 4 neurons are already are tuned. This is a fact which is experimental. And then what we want to understand is the other experimental fact that layer 2, 3 also, the neurons are tuned. And we, under the assumption of our hypothesis, okay? Under our hypothesis that the connection from layer 4 to layer 2, 3 are random, no functional organization. So why there is an issue here and what will be the solution? So the issue here comes in if you look at what is the structure of the excitation from layer 4 to layer 2, 3. And the observation that this excitation is weakly tuned to orientation. So our assumption again, we assume tuning in layer 4. So this is an equation which formalizes tuning. R of theta is a baseline plus a term which depends on the stimulus orientation theta and the preferred orientation of the neuron. This is for a specific neuron. And with a term which depends on the contrast. And again, random connectivity. What that means, that's random connectivity, it means that if I'm taking one neuron in the cortex, so it will, in the layer 2, 3, it will receive inputs from a bunch of neurons from layer 4 with different preferred orientations. Okay, they can also have different tuning weights, but I mean, for us, it's enough to consider the case of they're all the same tuning weights, but only preferred orientation is random. So if you compute now this input, so you sum up this input and you multiply it by the strengths of these connections, so it's very easy to see that due to the law of large number, so the mean over the stimulus orientation is large and in fact proportional to the number of inputs, to K, which is the number of inputs, and the average activity average over the stimulus orientation of layer 4 neurons. Now, if you zoom in, you have a small modulation with orientation. The point is that this modulation is very small compared to the mean, to the unmodulated part. This can be formalized by this equation here. So this is the thalamic excitation, the part which is non-modulated. And this is, again, this is because of the law of large number. Actually, you can say something about the amplitude of this modulation. And this is proportional to the square root of the number of inputs that this cortical neuron, that this layer 2, 3 neuron is receiving. So let's say that if you take K, which is 400, so this means that the modulated part will be 20 times smaller than the unmodulated part. All the information, all the information about the stimulus orientation is carried by this modulation. So the question is this information, can this information have an effect on the response of neurons in layer 4? In layer 2, 3, I'm sorry. Here we are from layer 4 to layer 2, 3. So this is what we investigated. And what we showed in this paper from 2012, we showed the following. I'm going to read it. I mean, this is a part which is rather technical, and I will not enter into the technique, the aspect which is technical in order to, which, I mean, if you want really to explain, understand at the conceptual level how it works, it's a talk by itself. And this is not, I mean, it's in the paper, it's published. So what we did, we showed that even though the total feed-forward and total recurrent excitatory and inhibitory inputs all have a very weak orientation selectivity, strong selectivity emerged in the normal spike responses, so in the output, if the network operates in the so-called balanced excitation inhibition regime. And this is because in this regime, the large untuned components in the excitatory and inhibitory contributions approximately cancel. As a result, the untuned part of the input into a neuron, as well as its modulation with orientation and time, all have a size comparable to the neuronal threshold, and this can, in the output, this can have an effect on the output of the neuron, since both are on the same order. So now we're going to read the qualitative understanding of what happens. I mean, it's a way, in fact, to reformulate what I have just said. Okay, so for a network to operate in the balanced regime, so that's the definition of the balanced regime. In fact, it has been a lot of work on this question. This was initiated by the work of Carl von Vreeswijk with Chaim Sampolinsky. So in this regime, the recurrent as well as the feed-forward inputs into the neurons have to be large compared with their threshold. And what is showed that what can be shown that under very general conditions, the balance between excitation and inhibition emerges from the recurrent dynamics of the network, so that the net input is much smaller than its excitatory and inhibitory components. And this is all the whole point here, is that this emerges without any tuning to guarantee this balance. And the reason why it emerged without tuning is because of the recurrent dynamics. Okay. So now, again, what happens if the network, our network, V1 network operates in the balance regime? So if a neuron receives its input from any cells with preferred orientations randomly distributed, homogeneously between 0 and 180 degrees, the orientation modulations of the total excitatory and total inhibitory inputs are small compared with their means, and taken separately, these inputs are weakly selective. However, when they are combined, the untuned part of the excitation and inhibition balance, the tuned part of the input is amplified, while the untuned part is suppressed. As a result, the mean and orientation modulation of the net input are of the same order, resulting in selective neuronal output. In fact, in the balance regime has a very interesting property. That is, there is amplification. In that regime, there is amplification of small effects, small fluctuations, for instance. Can be also temporal fluctuations. Here we are talking about fluctuations or modulation in terms of, with respect to stimulus features. But in fact, it's much more general. This is a very interesting property of the balance regime, which, as we show here, can have important functional consequences. Okay, so we know now that layer 2-3 can inherit orientation selectivity from layer 4. And now the question is, from where the orientation selectivity, how the orientations, from where, what is the origin of orientation selectivity in layer 4? So here now we are going to deal with the thalamus and layer 4, and we will ask the question, if we assume that in LGN, and this is something which is well accepted, plus some changes, a bit of other detail. So neurons in LGN are not tuned. So now we want to see how the selectivity can emerge. Again, under the hypothesis that all connectivity is random in layer 4. Okay, so the first issue, so we are going to do this the framework is the following. So we consider a model of LGN sending inputs to layer 4. The LGN is responding. The visual stimulus is a drifting grating characterized by spatial frequency and its orientation. And the question is again, so here we assume that we have a 2D network of conductance-based neurons in our model. LGN to layer 4 and recurrent connectivity at random and only distance dependent. And the thalamic excitation and layer 4 now this is the consequence of this type of visual stimulus will depend on time. So as graphically done in the experiments, so you take the response of a neuron, you expand it in Fourier, you will have the average and the first Fourier component, the F0 and the F1 component of the response. Important parameter here is the footprint of the connections between the LGN and the cortex. So, structure of thalamic excitation. So what can be shown analytically is the following. If we consider the thalamic excitation and we are interested in the selectivity of the F0 component, so this will be, the OSI will be 0. If, now for the F1 component, the selectivity depends only on one parameter, which is sigma over lambda, except a very weak dependence on the number of LGN inputs to a given cell. This number is K. So here you see the prediction, the result of the analytical calculation, which is this curve here, as a function of the ratio sigma over lambda. And you have the simulations here, the black and the red. The red is for, we assume K 50 or K is 100, and we compare it to the theory, which is done in the limit of an infinite number of inputs from LGN to neurons in cortex. So you see that already with K equal 50, you see you have a very good, it's a very good agreement with a theory which was open, in fact, of the fact that it was established in the limit K goes to infinity. So very robust. There is a peak, there is a maximum, and we're going to compare this curve with this data of the result of the theory with experiments. In fact, experiments has been done to measure this thalamic excitation and its property. So this was done in the group of Lin and Scanziani in 2013. They consider adult mouse at given spatial frequency. You will see why I am emphasizing this fact in the experiment later. So what they do, they essentially using optogenetics, they inactivate the excitatory neurons. They measure the activity, they measure the excitation that neurons in layer 4 receives from the LGN, because now when there is no, they measure the excitation, the excitatory current that the neuron in layer 4 has in presence of stimulus in two conditions. One, with suppression of the recurrent excitation, and one with keeping the recurrent excitation. So you suppress the recurrent excitation with optogenetics, you subtract, and then you can have access to the thalamic excitation from the LGN, and also you can compare it to the total and measures amplification and other things. So they did the experiments, and this is what they find. They find that the OSI for the F0 component is very small, 0.03. The SI for the F1 component is 0.23. So we can compare now with our theory. OSI of F0 is very small. In the theory would predict 0, but I'm not sure if it's 0.03, maybe it's 0 up to experimentally it's maybe 0, but compatible with 0. For the F1 is 0.23. Let's see what that means. If we compare, if we compare to our theory, it means that basically this is at the maximum. Okay, so in the experiment, what they measure, they will measure the effect is maximum. The selectivity of the F1 component is maximum. Again, the prediction of the theory, what the theory says, is very robust. It's purely geometry. So now if we compare again with the theory, so we can say something about sigma, about the footprint, and take into account that this was for 0.04 cycle per degree, and we arrive to number 6 degree, which correspond more or less something between 75 microns to 100 microns. Now they did something else. They also measured the on and off structure of the thalamic excitation. And what they found a lot of overlap between the on and off subregions. So we interpret this result, in fact, as a lack of organization. Okay? The on and off subregions are strongly overlapping. This is not what the standard Hubel and Wiesel view would say. In fact, the on and off subregions measured in cat and monkey are very well segregated. And there is an offset, and the typical offset, in average, would be around 5 degrees. What our theory says about that?
David, excuse me, 10 more minutes.
Okay, so I have really to run. So what our theory says about that? Our theory says that indeed we can reproduce this result. And reproduce this result with an offset on and off, which is compatible with the data. Okay, visual response properties in layer 4. So it works when we use this input in our model, in spite of the fact that there is no organization, we get orientation selectivity in layer 4. Of course, there is a lot of diversity, neurons which are more tuned, less tuned, neurons which are not tuned. And the distribution of the OSI is, I mean, this is the histogram in our model with a mean which is around 0.20, 22, 23. So this is the OSI of the response of the neuronal cortex. There is a correlation between the preferred orientation of the thalamic excitation and the preferred orientation of the response of the neuron. Now we have a prediction, in fact, which comes from the model. We convinced it was not completely easy, but in fact, Nicolas Privé and his groups are very, very enthusiastic when they do science. So eventually they tested the prediction. What is the prediction? The prediction is from the model that orientation preference changes with spatial frequency. You remember that for cat and monkeys, it was not the case. And in fact, this comes from the random organization in the model. And you have here 4 examples of neurons, some of them where the difference is big, there is a lot of change. So this is for 2 spatial frequencies. Some of them were it less. There are other changes also about the neurons which can be more tuned when the, I mean, change also in the OSI. Okay, so there is a lot of diversity, but the bottom line here, that there is this dependence, it change with spatial frequency, and this is also shown here, where we plot the preferred orientation at 0.03, 0.02, and 0.01 spatial frequency versus the preferred orientation at 0.04 cycle per degree. This is the model. What happens in reality? So this is, as I said, was measured by Javruti Patatkal in the lab of Nicolas Privé, using sinusoidal drifting grating with various spatial frequencies, doing calcium electrophysiology. The electrophysiology was intracellular as well as extracellular. And this is what they find. So they find indeed the decorrelation when you change the spatial frequency 0.02, 0.03, 0.02, 0.1, and you compare it with 0.04 cycle per degree, you see this decorrelation. You see this in the model here. You can compare it here. There is more decorrelation in the model, maybe, probably, but essentially, the prediction of the theory is very fast. Now, this can be summarized also in this figure by measuring the circular correlation of the preferred orientation for different spatial frequencies. So this is a correlation for 0.03 to 0.04, for the calcium in green, for the electrophysiology in blue, for our model, which I call circular model, which assumes that there is no orientation selectivity in LGN, so it corresponds to all what I've described until now. And also we can compare it in a case where you put some orientation selectivity in LGN. In fact, there is some evidence that in LGN of the mouse or the rodent, there is some orientation selectivity already in LGN. Bottom line, there is a quantitative agreement between the theory and the experiment. So we can also see this in another way by looking at the receptive fields in the Kx, Ky. Kx and Ky, this is the component, X component, and the Y component of the wave vector of the drifting grating. And what you see that when you plot the response as a function of Kx and Ky, is what is shown here. So the region where there is activity is not a line along the radial direction. This is from the model. This is correspond to this kind of shift in the preferred orientation for difference. So this is 0.01 and 0.04. And this is in the, I'm sorry, this is in reality. And this is the model. Membrane potential here, this is the tuning, this was intracellular. This is the KX-KY plot of the activity, and this is the model. So we call this intricate. So what that means? That means that in mouse, V1 already encode, or neuron in V1 already encode for more complex aspects of the stimuli. It's not, not separating the features, orientation, contrast, and spatial frequency, but also already responding to global properties of the stimulus. So this is in contrast with the monkey model. Now, I'm going to go quite fast now, although what I'm going to explain now is not published. So there was, after our paper of 2012 was published, so there is this study from the group of Flogel, published in Nature, and they said two very interesting things. Also, neural responses were already highly selective for visual stimuli at eye opening. Neurons responding to similar visual features were not yet preferentially connected, indicating that the emergence of feature selectivity does not depend on the precise arrangement of local synaptic connections. We are very happy when this paper was published in 2013, because essentially it means that our theory as such, as it is, can be compared to what happens before critical period. Now, after eye opening, so long time later, local connectivity reorganized extensively. More connections formed selectively between neurons with similar visual responses, and connections were eliminated between visual and responsive neurons, but the overall connectivity rate did not change. So that means that now, later on, there is, during development, there is apparition of selective interactions. How much? How many of them? So this thing is answered in a later paper. And the bottom line of this paper is that the number of such selective or feature-specific connections is small compared to those which are non-specific, and also they are stronger. Now, the theory should say, one, what can be the effect of this on the selectivity? What that means, what would be, in order to have an effect, what is few and what is strong, okay? And this is what we did. We studied this in, again, so this is a combination, this is not published. This is a combination of analytical calculation and simulations. I will just say the bottom line of that study, that indeed only a few specific connections can sharpen selectivity. So we showed this by assuming the model in which K non-specific connection, typically K will be 400, and square root of K specific connection, so 20. So 20 compared to 400. JEE is the strength of the non-specific connection. JE spec is kappa factor multiplied by the strength of the non-specific connection. So this is, kappa will be vary between 0 and something. And our question is, in this situation, we have here again, 400 versus 20. Is it possible to, what will happen if we increase kappa? So this is shown here in terms of the histogram of the OSI. So what you see is that the OSI for kappa 0 and kappa equal 4, the OSI moves or broaden, and the average is increasing from point here, 0.20, let's say 0.23, 0.24, to here, something which is close to 0.35. This is for kappa equal 4. And we plot here the function, how this population average OSI varies with kappa. And what you see that indeed there is an increase in the selectivity, degree of selectivity. What is very important is two things. One, this story shows this happens only if the cortical network operates in the balanced EI state, and the specific connection are sufficiently strong, but much smaller than square root of K, much smaller. So it means that this amplification, the strength, this kappa is much smaller than square root of K. So it means that these connections are strong, but not that strong. So we have a small number of specific connections, stronger, the specific connections are stronger, But kappa is smaller than square root of k, typically, yes? Square root of k is 20 here, it should be 4 or 5. Of course, if kappa was what you need, you would need that kappa to be very large and going, I mean, varying also with k, increasing with k. So then, I mean, in a way, it will be obvious, yes? But the point which is here very important is that the interactions have to be stronger, the specific connection have to be stronger, but not that stronger. Now we can compare this to data because it has been studied. So how the orientation selectivity index increases along development has been studied in 2015 by Neil and Stryker. And what you see that it goes from 0.2 to 0.4, 3.5. And this can be explained by our theory. Conclusion, I call this conclusion the power of randomness. So now I'm going to go in the reverse order, I think the talk. For the thalamic excitation, the observed orientation tunings is what our theory shows. And the receptive field properties can be accounted for with random LGN to layer 4 projections. The thalamic excitation can give rise to orientation selectivity in layer 4. Layer 2-3 can then inherit orientation selectivity from layer 4. We have a prediction, which is verified. The preferred orientation of V1 responses changes with spatial frequency. There is intricate 2D orientation spatial frequency receptive fields. And finally, as a consequence of the EI balance, a few strong specific connections can sharpen orientation selectivity. What I did not show here is that we can also explain direction selectivity if we assume that also we have a diversity in the dynamics in the LGN. So my final acknowledgements. So the people who participated to this collaboration: Carl van Vreeswijk, Roman Matteau, Jagruti, Nicolas Privé, the other people that collaborators, Jonathan Levenstein, that I would like to thank very much for hosting me in his lab as visiting professor of the university. These are the grants. And I would like to finish by advertising the worldwide theoretical neuroscience seminar that we are running with Carl van Vreeswijk every Wednesday since the beginning of November, and that will continue even after we are beyond the COVID episode. Thank you very much.
Thank you, David. Before we move to the question, I'd like to apologize. We had electricity failure here in ELSC, so some of us missed some of the talk.
I realized it.
So let's move to the questions. You're welcome to ask. Yoram?
Yes. I actually have two questions. So the first one
wait,
is...
wait, I would like to see you. I would like to see you. Ah, I see you. Yes, good. Yes.
The first one is about transformation from layer 4 to layer 2-3.
Yes.
So just about this idea that you have many neurons that are tuned a different manner and you combine them, but the mean is subtracted. So at least naively, you might think that noise would not be subtracted and you will somehow amplify and degrade the signal to noise ratio. And I wonder if you can say something about that. And but the second question is much more broad, and which is that the whole work that you presented is about the mechanisms and the network architecture. And somewhat connected to my first question, did you or others think in this context of balance framework about the information processing features of the network, and whether the organization of the network is beneficial from this point of view?
Okay, so regarding the first question, I don't think that this will happen. I mean, in fact, it's taken into account in all the theory, this noise. Mm-hmm. Yes, it's inherent also because of the balance of excitation inhibition. Yeah, so it's already there. It's taken into account. Yes, so we estimate this effect. We estimate the effect of the heterogeneities in the connectivity. Yes, and we estimate the effect of the noise. So this is taken into account. Okay, this is one thing. Now, the second point, I mean, it's always, this is a question that is always, I mean, frequently asked. So what are the functional consequences of this? So some people would say, okay, so you are working in mouse, but mouse, they don't see well. So maybe it's connected to the fact that there is no map. And maybe it's connected to the fact that you have this randomness. So for this, I would answer in two ways. One, orientation selectivity in the mouse, in fact, eventually is very similar. The degree of orientation selectivity is very similar to the degree of orientation selectivity that you have in cat and monkeys, at least according to the experiments of as a result of Neil and Stryker. There is a kind of a debate here. I mean, you have other experiments saying that it's a bit less. But I mean, bottom line here is that there is orientation selectivity, which is substantial. This is the first thing. So there is, I mean, so there is processing of something, yes? Now, the processing, what I've said here, is basically that it's different from what happens in cat and monkeys. In cat and monkeys, the idea is, and this is the standard idea, that in V1, there is analysis separation of the contribution of the different aspects, of different features, which are present in the visual scene. Yes, so for instance, if you have a natural image, yes, so V1 is going to decompose, yes, in orientation, in velocity, in contrast, in spatial frequencies, and all these things, yes? What happens here, and then it's later on in the visual pathway that all this is going to be combined to give rise to the percept. What we say here is the key result here is that we claim, we argue, actually we argue that this is not what happens in the mouse, that already in V1 process the information at a more global level. So here we showed it for spatial frequency. We can show it also for the contrast. There will be, the contrast invariance is going to be broken. Okay, maybe for the color. Okay, so everything will be important. Now there is another thing that people says, yeah, but the mouse, you know, it's again, as I said, it's not a visual animal. So maybe this salt and pepper is related to that. The answer is that squirrel have also a salt and pepper organization, and it would be better for them to see well than to see badly. Otherwise, would not be able to jump from one tree to the other. So did I answer your question?
Well, about the first question about the noise, I don't mean the noise in the connectivity and the internally, the noise that is generated internally in the network due to the balanced state. I'm thinking more about the fact that the inputs to the network might be noisy. Okay. And there it's not less clear to me.
Yeah, it's true. It's also less clear to me. Yeah.
More questions? Okay, so I think we'll thank you here, David, for this nice talk. And we'll see you all here next week for the next seminar.